A little bluestem is important because that's one of the major prairie grasses in the tallgrass prairie region. It is on our target list as being very important for habitat restoration. So that one in particular is one we would like to seed bank. Little bluestem is on our target list for the restoration collection. It is highlighted in light blue, which means that we want to collect this particular species along maternal lines. We need to collect each individual maternal plant and bag it separately. So we need at least 50 collections. Okay, we're going to begin the collection. First, we're going to collect along maternal lines, which means we're going to make 50 collections of individual plants. What we want to do is collect the seeds off of each individual plant and put it into a separate bag. I'm going to carry this canvas bag to hold it all. This works much better with two people. One person can hold the bag and store them while the other's collecting, but I'm going to give it a shot on my own here. Here's one individual plant. We strip it. Not all of the seed comes off. You don't want to take all of it. You want to take about 20%. You put it in the bag, fold it over. That's one collection. Do the rest of the collection like we would any other seed collection, which is just to bulk it all in one bag. So I have a nice grocery bag with handles here, and we just wander around the population and strip a portion, about 20% of the plant. And as you go, make sure you count. It's important we get a count of how many individuals we've. So I keep that in mind as I'm moving around. Okay, we need to collect a sample of a plant for an herbarium specimen. It's best to collect the entire plant all the way to the root. You can fold the plant because it needs to be folded to fit on an herbarium sheet anyway. So I fold the plant, place it in the bag. Eventually we'll take this to back to the vehicle and press it in the plant press. We want 2 specimens. Next we need a leaf sample as a DNA specimen for future analysis in the lab. We have a baggie full of silica gel envelope inside. We find a real green leaf if you can. In the fall sometimes that's hard to do, but here's a nice green leaf at the base of this plant. Fold that up. Put it into the envelope. Seal it. Then there's a little bit of data that needs to be recorded on the outside of the envelope. The name of the plant, date collected, who collected it, and the location. And then that goes into the plastic bag. The silica gel dries the specimen down and eventually goes into a freezer. We're back from the field, and we need to take the specimens we collected for the herbarium and put them into this plant press. Just take regular old newsprint. Take each specimen, which we folded to put in the bag, which is fine because you got to fold it to put it into the press. Make sure you get the whole thing in. Fold it. Let's be sure to record the plant name, date, and your name on the outside edge of the newsprint. And you want to make sure you cinch the straps down tight. It's good to go. We're here at the Dixon Seed Preparation Lab at the Chicago Botanic Gardens Plant Conservation Science Center. The first step after bringing seeds in from the field is to give the seed collection a collection number, and that's through our database. This collection number for little Bluestem is going to be 1622. After we give it a collection number, it goes into the seed dryer. We need to dry seeds down to 15% humidity before they can be frozen in the seed vault. So seeds need to be placed in this seed dryer for about 3 weeks. The next step in the seed banking process is to clean the seeds. We take them out of the bag, dump them into a pan so we can see what we got. This seed is fairly clean, but it does have some extraneous plant parts, stems and leaves, so we have to remove those by hand, at least the major plant parts. And then we get down to just the seeds. We put them on a sieve, because each of the florets are kind of grouped together. It makes it easier to put it on a sieve and just kind of push them through. It's important that the sieve size is large enough so the seeds easily pass through. Otherwise you have a tendency to damage the seed by grinding them through a sieve that's not large enough. We use these rubber stoppers that help push it through, and this separates the seeds. Because little Bluestem is a very fluffy seed, this particular instrument, our counter, will not actually do a very good count. The seeds don't pass through the electric eye, and so what we do instead is weigh the seeds. When we're done counting and weighing, the seeds are then divided. Some of the seeds go to other seed banks as redundant storage, and the seed that remains here is eventually put into these foil packets, airtight foil packets. They're sealed and they go into the freezer. After the seed is sealed in these foil envelopes, they're ready to be stored into the seed vault. We keep our freezer at -20 Celsius, which is about -4 Fahrenheit. So we have to dress appropriately to go into the freezer. If seeds are dried to 15% humidity and stored at -20 degrees Celsius, they can remain viable for long periods of time.